happy to be able to introduce Tom Carroll. Many of you know that Rockefeller has been sort of thinking about how to support and centralize bioinformatics for quite a while. So I arrived here about 2 years ago, and it was one of my top priorities. And it's taken a while, but we hope it's finally worth the wait. Tom comes to us from the UK. He was born in London and did his undergrad in genetics at Swansea University. He did his master's in bioinformatics at Exeter University and his PhD in toxicogenomics at King's College. While he was at King's, he won the Roger Gilbert Prize in Quantitative Genetics and the King's College PhD Symposium Prize in Bioinformatics. Tom did his postdoc at the MRC Clinical Sciences Center at Imperial College London, where he worked with several groups and gained experience in many different types of data, including epigenetic and Rna-Seq. After that, Tom joined the Cancer Research UK program as Senior Bioinformatics Analyst, where he participated in a lot of downstream data analysis and training development. Then finally went back to MRC London at the London Institute of Medical Sciences as head of bioinformatics, where he founded the first bioinformatics core at MRC. Following that, we were lucky enough to steal him here after a long and dynamic recruitment process. And Tom started here as Rockefeller's first head of bioinformatics in June of this year. And this is really his sort of grand unveiling. And although he's not been for lack of work recently, he's really finally getting the center off the ground, and he's going to tell you all about his plans in a moment. Just to brag about Tom a little bit, he is an accomplished researcher in his own right with nearly 40 papers in peer-reviewed journals. And he's also written one of the top 10 ChIP-seq QC packages called ChIP-QC as part of the our bioconductor framework. Top 10 most downloaded ChIP packages. So we're very happy to have him. And with that, Tom.
Great, thank you very much, Anthony. Great, so thank you very much for coming. As Anthony said, I'm Thomas Carroll. I'm the new head of bioinformatics for the Rockefeller University, and that means I direct the Bioinformatics Resource Center here. And it's that what I want to talk about today. I'm going to give you a little bit of an introduction to the BRC. So to do this, I'm going to start off by talking a little bit about who we are, some of our experience. I'm then going to talk about what I believe our role here at Rockefeller is, and then I'm going to talk about some of the work which we are doing right now and into the future to try and meet this role. So developing pipelines, putting together some training, and of course, data analysis. So, as Anthony just said, we're a very new core. We've only formed in the last 3 months, starting in 2017, July. So this is meant to be a centralized, shared resource where anyone within the Rockefeller University could come and seek us out and ask for bioinformatics support, advice, or analysis. So I say we a lot during this talk, but it's actually just me at the moment. So we'll say the royal we, or as my friend at the back will say, the majestic we. But we are recruiting, so right now I'm trying to recruit for a bioinformatician and analyst to come join me, hopefully have this position filled by the end of the year, and in 2018, at the very beginning, we're hoping to hire a second analyst. The reason we've staggered this a bit is to try and see what the work is at Rockefeller, what we really need, and then I can target the hiring of a position to fill these skills which we require. If you want to find us physically, we're in the IT pavilion. I'm tucked behind the help desks in the office next to Anthony. And I'm going to say we are working, or we will be working closely with the Rockefeller University Hospital team to try and consolidate some of our workflows and also ensure that we're not duplicating effort in anything we're doing here at the Rockefeller University or in the hospital. So I'm now just going to talk a bit about myself, because it is just me, remember, some of my experience. As Anthony said, I've come from wet lab biology originally and moving into array analysis as part of my PhD. And from there and in my positions in different cores, I've learned a lot about array analysis and moving into next generation sequencing data of all different flavors, including things like transcriptomics, but also single cell transcriptomics and then quantitative epigenetics defining large marks across genomes, all sorts of different types of NGS analysis. I quite like to develop software when I feel it's needed. I work in R, Python, and JavaScript mostly. So I put together a nice JavaScript library when I was in CRUK, and this was meant to be a simple way, just within a browser with no dependencies, of aggregating all these quality metrics we get out of different tools, and then being able to visualize them, query them, interact with them. Written a number of pipelines, including the CRUK next generation sequencing pipelines and the pipelines I put into the MRC, and I wrote these all in Python. It's a fantastic language to wrap up your workflows in. But I'm a big fan of, and usually I would be wearing a I Love R t-shirt. Or I Conduct R. I think this is a fantastic repository community to contribute to when you can, certainly to take part of when you can. So over the years we've put together a few packages, mostly around this type of role, so quality control and visualization of different types of data. Sorry, what was that?
The mic is off after you gestured about the R t-shirt. You're watching.
Wow, people are paying attention. Am I still on? Great. So, also been involved in training over the years. I think it's really useful and really important to have training within an institute to try and disseminate some of the methods we use. And over time I've built up quite a repository of courses where I will present this later today. This ranges from just getting an understanding of what the data types we have are, so what type of results you might be getting out of a pipeline, to doing some more complex analysis yourself.
I'll charge for frisking.
Thank you. Great. I'll try and just leave that there. So on top of some of the training I put together myself in CRUK and in the MRC, I've also worked with this guy here, Mark Dunning, who's in CRUK Cambridge. He's the head of training there.
Jeff, that won't work in this room. I'm going to change it then. Sorry.
I can hear you.
Okay, so I will try and speak a little bit louder then. So working with Mark, we've actually built up a nice repository of shared material. So this is where we can pool our courses, our repositories of what we want to present, and then we can avoid doing things like duplicating work, producing courses which we want to present in individual universities. And this is a nice publicly available repository now, and although we started it, we've found a number of people. So that's a little bit about my background. Now I'll talk a little bit about what I think our role at Rockefeller is, and I think it's quite a general remit, is to support bioinformatics analysis anywhere from project conception through to hopeful publication of the actual data. Okay. So that will involve things like experimental design. We hope that if you're going to run an experiment and come work with us for analysis, you'll come talk early to us about experimental design. But also we may be involved in developing new methods, new software fitted to your particular question, or even, or especially, analysis, processing, and delivery of data. And I think that's often our largest part of our work. So just speaking to that analysis now, I was thinking about what type of analysis we will be doing, and I found a really interesting paper from a few years ago which kind of summarizes the different types of work coming into a research core here at the University of Texas over 18 months. And I think what's really clear is we have mostly two components, but we have work which this is on the Y is frequency of a method, and along the X is the different methods. We have some methods which we will do a lot. So we will repeatedly do the same type of analysis, and this is often pre-processing or processing of data, or some standard downstream analysis, differential expression, peak calling, motif hunting, something like this. On the right here, we actually have quite a bulk of work, which is unique. We only do this once, and that will often be something very tailored to a particular question, often just prior to publication. And I think this is actually a fair distribution of the type of work which I see in the cores I've run, but also I think across all cores, where we will have things we do every day and things we do just once. So we need to be able to support things like processing, pre-processing of data, standard downstream analysis, but also expecting to do a fair degree of custom analysis. In this work, I want to make sure that everything the BRC does is going to be able to support for the long term, because projects can go on for years before they're published. And we want to make sure we're using the best practices, obviously, but we do, and I'm very keen on this, is make sure we provide documentation for every analysis we do, and that I'm ensuring what I do is reproducible. And I'm just going to speak about that for a couple of slides. I just want to point out before anyone, these are the good guys. I put this up and people saying they might be the bad people. So irreproducibility and bioinformatics can come about from some very easy, innocuous sources. So we can have things like just not documenting the type of software you used, what the version was, can often make it hard to reproduce an analysis. And this can be as bad as coming to a paper and they've written, we did the analysis in custom scripts with no further explanation. Or it can be something like this, where you have a simple copying or coding error early in your analysis, it's missed, and it will propagate downstream to the final results. And that's actually what this is about here, and I think this is a good story in some ways. So these two here are what we call forensic bioinformaticians, and what had happened is in their analysis, they'd shifted the row IDs by 1, so every ID was matched wrong to a gene, but this continued all the way to a drug trial before anyone actually spotted it. Okay? So from something really small, which I actually don't think you would ever be able to spot, because it's so early in pre-processing, propagated into something really awful, and we had to stop a drug trial. They had to stop a drug trial at that point. On top of potential errors in your analysis or problems like that, I believe it will become a requirement for publication in major journals to submit some links, some description of your code alongside the results which they were generated, or which generated. Already you have to submit data when you try and publish a paper. If you look at the author instructions in places like Nature, they are saying and they're suggesting that we want to have links to your code. So I think it's only a matter of time that it becomes a requirement that when you publish, you need to explain exactly what you did. So, hopefully to try and ensure that we are reproducible and that we can deal with this large number of work which you do more than once, processing, standard analysis, I think it's a really good idea to try and centralize our work into trying to find or build workflows which can automate or optimize the processing of really common data types out of high throughput biology. So this should help us by building a pipeline to process this, reduce things like copying and coding errors, allow us at that point to capture all these versions which we require, but also hopefully accelerate the analysis and allow us to give back data a little bit quicker to the user. On top of that, I really like workflows because I think once you start looking at data the same way every time, even if you need to process it differently in the end for your paper, you start to get a real good understanding of what these characteristics, what these metrics around the data really mean. We can pool our resources, and once you have a workflow in place, it makes it very easy to start to test out new methods, something a bit more experimental. I already have my workflow, and I can change something and compare that to the old results. So thinking about what workflows I need to build, I think one of the most common data types we have, or certainly coming to me, is next generation sequencing. And in that, I think some of the most popular methods are RNA-Seq, ChIP-seq, and ATAC-seq. So that's really what I'm targeting. And I want to build a pipeline which can retrieve and process the data, perform some basic quality control of the data, and maybe a default analysis, a pairwise comparison between groups. And all this I want to wrap up in a nice deliverable report, summarizing some of the interesting metrics, characteristics of the data, some of the output from the analysis. And I need this to be scalable from small data sets to huge data sets. We'll see now we've got a single cell system, right? But also I need it to be reproducible. Okay, so I want all this to be absolutely reproducible and all my versions to be controlled, both for the software but also the genomic data I use in processing this. So what I've put together so far is a R package. So I've moved away from using Python to wrap up our workflow. Using an R package I call the NGS Piper package. And this can analyze Rna-Seq, ChIP-seq, and ATAC-seq data. What's a really nice feature of it is because it's all in R, I can actually use Bioconductor and I can get all the genome annotation I need for at least 17 model organisms without having to download any other data. I can obviously allow the user to supply their own data. I get version control through PackRat. I get the ability to scale from my laptop, and all the examples I'll show will run on my laptop, to a high-performance computing environment by using some of the abstraction layers afforded to me in Bioconductor. I can get testing because it's an R package, it's easy to test it. And R has some fantastic tools for things like dynamic reporting, and I want to take advantage of all that too. So I think it's a nice choice to use R here, but I'll be happy to debate this at the end. So this is an overview of an RNA-seq analysis, and I just wanted to give you an idea about how it works. So this is the RNA-seq kind of workflow in the pipeline, and it's very simple really. On the left, we take FASTQ, we copy that locally, I then clean this up, so I remove adapter contamination, poor quality reads. This comes into the main part where I will take a user-supplied sample sheet which contains the groups, the metadata, which you think is interesting. And then this will follow on the left, a standard processing. I'll do some alignment. I will count in genes or exons, gain some statistics from this. I'll make the bigwig the thing we can look at in a genome browser, and then I will output all this. On the right-hand side, we have something a bit more advanced. So these are some methods which are being published, the one I used just this year, but it's well tested. And this is using pseudo-alignment or k-counting, where I can go straight from the FASTQ to actual counts on genes, counts on transcripts. And this is ridiculously fast. And from there, I can do differential gene expression, transcript usage, and I can do some gene ontology enrichment. I think a really nice feature of the pipeline is, as I said, you can get all your... Everything's running out of batteries today. I got two, it's okay. So you can get your data from, you can get the genomic data you need from Bioconductor for 17 model organisms, most organisms you're working with. But if you are working on some avian species, for instance, you can supply the genome faster, the gene models, and some ID to symbols ID to go. And then the pipeline itself will then prepare any additional annotation we need. So this is things like making the transcript sequences from a combination of the FASTA and the GTF, or flattening our GTF in order to do exon counting. Right, so you can give it minimal annotation and it will do a lot of the work then to try and build all the annotation we need. So so now we're just going to concentrate, after all this, actually, all these results and all this processing, it then produces a little report. So it produces a directory which contains a report and all your output files. Okay, and there's actually this bit here on the report which is generated, I wanted to show you now. So the results files it gets, these are simple things like sequences, after trimming, the aligned data. If you want that, I will give that back to you. If not, I will keep them. I will always give you back some normalized signal graphs. So this is something where you can actually compare between your samples. It's already normalized the total number of reads. And I will give you things like counts in genes and transcripts and exons. And the idea is that you might want to go on from there and do some of your own analysis with this. And I give you some default analysis, and I just run, based on your metadata, all pairwise comparisons. And we'll get differential gene expression, differential transcript usage, and some gene ontology enrichment. What I want to make sure I give you, though, is a report, and this is what I call a dynamic document. So this is generated the same time as your results are generated. So it's very important to have that. And this describes the analysis performed, what the input and output files were, what the versions used, right? So this makes sure that we're reproducible with a combination of this and this. And it summarizes up some of the QC and some of the analysis in tables or some various plots. So I'm now going to start moving into the live demo part. So forgive me if it doesn't work. Right, so this is what the PDF output is. So you get 3 different reports back, actually, and they're all they're all the same, but they're in slightly different formats. So the PDF report, and I've got one open here, looks like this. So we have the name of the project, which is kind of useful. I have the date which I ran it, and then I have at the beginning here some QC in terms of alignment metrics, simple things like percent mapping, whether it's compatible fragment ratios is telling me whether I'm on the right strand, which is actually quite important. My mapping rates from standard alignment. I get some little plots which give me an overview, so if something's gone wrong here, that all the reads seem to be in one replicate versus another. And we have little things on paired end moving through to mapping quality, and then metrics on the counting, how many landed in reads, how many are outside. And then finally, I have one metric so far for Rna-Seq quality, which is the mean profile of signal over a gene. This can often be useful. If I see a lot of signal at the beginning of a gene, I might think I have some problems, some degradation. If I'm doing Ribo-Seq, I do expect to see a lot of signal at the beginning of the gene because I'm not picking up untranslated regions. So this is kind of useful for Rna-Seq analysis and QC. And then run some default analysis, things like correlation between samples, using a heat map, trying to show you the greatest sources of variation and how your data separates across it. And then just a summary of the number of differentially expressed genes. So we have this many up, this many down, this many up under a fold change cutoff, this many down. At the bottom I have essential things. So I have the FASTQ, which went in, matching that up with, and I call it a genomics ID, but it's just a unique ID. And I also have the metadata, which I used in this experiment. So genomics ID, and I have the cell line. Okay. Beyond that, importantly, I capture all the versions I need. So now I know I'm using this version of the pipeline, 0.63. We've had a lot of iterations. But also I have, importantly, captured all the other software I use. Okay. So this report in itself should be quite reproducible. I know the input files, the output files, and I know all the software versions I used. The real point of this report is that it's self-contained. You should be able to print this, put yours on your desk, take it to a meeting, and you can quickly flick through it and get an idea about whether your experiments worked, how your samples group, just a simple overview. On top of the PDF report, I also generate from the same document the Word report. So this is all the same information, but put now into Word. So that means that you can take out the tables and do your own plots with them. It's a simple freebie we get from using this type of system. But what I was really excited about to do for the report is try and build some type of HTML report, which perhaps I could build into a web page. By adding JavaScript, I should be able to add some interaction. And actually, through using HTML and ports, I should be able to interact with some other programs. Okay. One program I really want to be able to interact with is IGV. For anyone who doesn't know, IGV is a tool which allows us to look at genomics data in the linear context of a genome. And it's very easy to use. Here I can just load in my files. It will find some nice way of presenting them, scaling all these samples individually though. But it is a useful, quick tool. But there is the options, if you can be bothered to put the formats together, of creating a lot of customization inside IGV. I can actually pass over sample metadata. I should be able to control the scaling of all these, put colors in if I needed. So I think I would like to be able to do this. So now I'm going to show you the interactive report. Let's hope this works. So first page of the report, and this is the web page which comes with the PDFs. We have multiple tabs. First thing I show you is the overview. We had this before, just to give you an idea about what's happened to your data. Tell you all the output files which you should have. So this describes what you should be looking for. You have obviously the reports, have the signal graphs, and then how the differential expression is named and the gene ontology is named. We have the same data we had in the PDF, but now it's got some interaction to it because I'm using HTML. So I can do simple things like filter these down or order them, like so. Just from using the same code, but now adding these interactive elements. And we have all the same pages. This is, again, the quality metrics coming from the alignment metrics here, pseudo mappability, and all these can be filtered or sorted. I think this is quite important, is that I've tried to make it consistently looking between the HTML report and the PDF. So if we go back to the PDF for a second, we had our plot just showing the mapping rates for this. And I should have been able to get a very similar plot here. But now this plot has some interactivity. So if I put my cursor over it, I will tell me what the actual counts are within the thing and what sample I am. I can scroll around a bit if I really wanted to. I can remove elements from it, and then I can take a picture and export that to show somewhere else. Okay. So all the way through these reports, I'm trying to make sure that the plots I have in the HTML match what I put into the PDF documents. As we go down, we have things on multi-mapping quality. And we have the same plot, which was the average distribution. It's not perfect, kind of overhangs here. But now I have some interactive elements to it. And I can remove things, etc. Okay, so that's the advantage of a bit of HTML to give us some interaction. One thing we have which is new here, which we can't have in the PDF, is this ability to load data automatically into IGV. So I'm going to try and demo this. Forgive me if it doesn't work. So here I have the equivalent of the sample sheet the user would have given me. So sample name, any metadata would be here. And I have a link which will allow me to load this into IGV auto-magically. So if I click that, it's loading this all into IGV. I don't need to know where the files are. It just looks them up and puts them in. Carries over some interesting information. So it carries over the metadata I wanted, so things like cell line. Actually, I'm giving away something later on in the talk. Let's get out of it. Cell line, and also I can set a group where I want them all to be scaled the same way. The real advantage of carrying over this metadata is now, when I start to try and organize my tracks, I can do things like sort, filter by metadata, or for the Rna-Seq, most usefully, I can actually do some grouping. So I can start to put all my samples in one sample group together, and then I can be able to compare them directly when I come to looking at genes of interest. Okay. So we have last page on the report, which is the differential expression. So again, we have the same plots, but now with some interaction on them. I can zoom in as well, but I'll show that a bit later. I have my principal component analysis plot, which is also able to scroll or move around. Most usefully, I can actually put my point over and know what that point is now without having to label it. And again, I can export this. Finally, we have this table at the bottom, which again previously reported our numbers of differentially expressed genes. But now I have a link, and I can open this up. And that should take me to a page which summarizes some of the differential expression results for that particular comparison. Right? So I've run all pairwise comparisons, and this is only one, right? Comparing the GM versus the HeLa cell line. And this is just an overview of your results. So here, for instance, at the beginning, I give you a heat map of the differentially expressed genes. Okay, so it gives you a nice static heat map just to show you any patterns you might identify here. Just because it's one group versus one group, it's not particularly interesting. What I haven't done is included any sample or row names, because often it's very hard to see gene names here. So what I'm trying to work on is an interactive version of this, where hopefully I can zoom in a little bit and start to actually look at some of these genes, and then I can scroll around them if I really want to. And actually they are searchable as well, so I can search for genes within there. Okay, so that's our interactive heat map. Also, we have this standard kind of plot, which is a volcano plot. Let me get rid of that interactive heat map. It's taking up memory. We have a volcano plot, so this is just the significance versus the fold change. Here I'm just plotting significant genes. Again, this plot you would often label them. Here I can just put my cursor over and know what the gene is. Okay, so I know this is MX1, I know this is exist. That makes it a little bit easier to identify and work with some of this data. I give you a differential expression table. This is just the output from DSeq2. But now, because it's HTML, I can do things like search for my favorite gene, MX1. It's not actually my favorite gene. But also, this is more experimental. We can hook up, we can actually link this plot to this table, such that if I want to select a point here, I can now see where it is in the table. It colors it differently. It gets cooler. We can go the other way as well. Where I can select some points here, and that's automatically filters my table down to just those points. Okay? So that's just one plot we've added so far, but we could obviously build this up a bit. Now I have my table filtered down to my genes of interest. I think this is a request from one of my users. I can add a link, and it should take me to the counteract telling me off for not installing that. But it'll take me to gene cards. And similarly, I have another link here, which does something really nice, where if I actually want to eyeball this gene inside the browser, I can just click there and it takes me to that gene. Okay, so I can actually look at what the gene expression looks like for particular genes as I work through my table. That's really useful for, ChIP-seq as well. And I think it's neat that we've managed to set this such that the scaling is always appropriate. These are already normalized, but now I can flick between a gene which isn't particularly well expressed to a gene which is very well expressed, and it will be able to scale the data appropriately to make them comparable. Okay. We also have, after that, some gene ontology enrichment of this. So again, I have a link here which will take you to Amigo, where you can get some knowledge about what this gene ontology term actually means. But I try and I think gene ontology is often complaints that it can be too general, too specific. So I've tried to deal with that a little bit better by offering some mechanisms here where if you sort by this column, you get something more specific. And if you sort by this column, you should get something more general. Right? So we're trying to correct for the Go graph in our gene ontology enrichment as well. Finally, we have, and this is the most experimental part probably in the pipeline, we have our differential transcript usage. This is using DEXeq, but also a method published just last month to try and identify genes which aren't transcripts, which aren't changing necessarily themselves, but the usage of them is changing within a change in gene expression. Okay, again, you add the gene cards, and I think this is actually where IGV starts to become really useful. Let me open up here. Because if I want to look to see how much I believe some of these changes, I can go to this gene. And this is meant to be differentially transcribed, but if I actually look at the end, or the beginning actually, you can see here, for instance, HeLa is only using, HeLa's the one which is using these first two exons. It doesn't seem to be used at all for the GM cell line. Okay. So I think that's a really nice feature of being able to interact with IGV straight from this report. Okay, I think that's everything I'm going to talk about with the report. Oh, that's the wrong place. Another freebie we get from writing this as an R package is we can actually build a web page straight from the package structure itself. So we have a nice web page there which will describe a little bit now, not what the pipeline necessarily outputs, but how to use it. So it shows you just the front page, how to install it. I have the manual so far for the Rna-Seq, but I'm including them now for ChIP-seq and ATAC-seq. Again, you've seen this overview already, but then I actually do have a vignette which describes what the inputs are and how you can run this and how you can use this locally on a laptop and how you can use this on the high-performance cluster. Another nice feature is we have this reference for all the functions we use, and over time I'm trying to export the functions so that even if you're not wanting to run the whole pipeline, if you want to collaborate and use this, you can come in and use some of the methods I've done. Tom Align, that doesn't look real. You can use some of the methods I put together in order to align the data, for instance. Okay. Something I am making sure I build into my systems is the ability to be able to test my pipeline, right? So I worry that because I use a lot of academic software, maybe I'm targeting academic software particularly there, but I'm using lots of other people's software. I don't necessarily know that this will work all the time. They might introduce changes. I might change my pipeline and break something which I didn't notice. So it's important I have testing built in. With R, as I said, we get some great testing, but we also can use Travis and GitHub to go every time I make a commit, it's tested, and if that commit is successful, it will then update the main repository of the code, right? So it's only then, once I am happy that everything does exactly what it did yesterday, it will commit and I will have something I can download. Another benefit of using this in combination with this tool is that every time I make a change to the reference manual or the vignette, my package is rebuilt and then the website is automatically updated. Right? So this should save us time and allow us to concentrate on doing more and more interesting things. So that's all I was going to talk about for the pipeline. Talk a little bit about custom analysis, because obviously workflows never get us all the way to the results we want. And typically we will have to spend some time either with those results or generating new results to go downstream and find some method which is particularly tailored to the biological question. So I've had already a number of projects I'm working on inside of Rockefeller, which I would consider reasonably unique, and this ranged from methods for doing unsupervised clustering using non-negative matrix factorization, which is a novel method for me, single sample gene ontology enrichment, and we could use this to look at changes of particular functional categories over time, and also things like differential periodicity. So this is an interesting project for me where someone had already discovered the periodic genes, but they weren't sure how to find the differentially periodic genes. So I can work with him and try and find some methods, and here we've managed to identify some which seem to have gained periodicity in the treatment. In all this analysis, as I said before, I need to make sure I'm reproducible. So when I give data back to people, I make sure I wrap it up in something like an R Markdown. Then I can present the actual results alongside the code, and also capture the versions. And this means that every analysis I give to someone, once they have that report and the document, it should be reproducible. Also, hopefully has the added benefit of, if I can put the result there and the code to generate it there, maybe encouraging people to look at the code and perhaps start to iterate or recreate versions of this plot themselves. It's important for us as a core in the BRC to find this balance between when we're doing analysis for groups, when we start to spot something which is either useful to multiple groups or is being used by multiple groups, maybe we want to take this method, wrap it up into a function, and put it back into our pipelines. Okay, if it starts to look useful for multiple people, maybe we can take the effort off doing the custom analysis and put it into a pipeline as an option or as a default which people will receive. Similarly, if we find that we're doing a lot of custom analysis which is parameter iteration, and perhaps it would be good to help the user do this themselves and we can help them with other parts of their analysis, we might want to start to do some training around that. And that's the last part of what I was going to talk about, is the bioinformatics training. So as I said before, I think bioinformatics training is very useful for an institute, and I think it's essential to show people at least the type of work we're doing, what it means. And we really need to make sure we cater for multiple levels of training, and this can be from people who just want to know what these files are, which I'm giving back to them, to maybe people who want to be able to pick up some of these processed results and do a little bit of their own analysis, to those who totally want to ignore what I'm doing in terms of a pipeline, and they want to develop the skills themselves right from scratch and be able to do an in-depth analysis themselves. Okay? So I want to try and cater to all of this. And to do so, over the years, I've tried to build up our training to be reasonably progressive. And I mean by that, you won't come across a method in something in Rna-Seq analysis course which we haven't already shown you in earlier courses. Okay? So we make sure that all the methods are contained within our course set. And we also try and make sure we can do this for the data too. So if you're coming across a particular data type in RNA-seq analysis, we hope we've shown you how to align it in the alignment, right? So we make sure that we can do this. This is a nice example here where we're doing sashimi plots, and this is one of our final courses, making sashimi plots. This is splicing plots. But we also showed you earlier on how to do that in a nice GUI as well. One thing I think is really important to do for training and isn't done enough is to make sure we can use real data in our training. So I don't want to use toy example data where you have part of chromosome 1 and you do your analysis of that, because there's no way a reflection of a real data analysis. You come across a whole different set of problems. So one way that I've set about trying to do this is to try and put our training in context. Given that publications should be reproducible, all the data in them should be in public repositories, and we hope that the methods there should be reasonably well described. So what I try and do is build my courses around published data, so that we show you how to retrieve the data, show you how to try and recreate some of these results within the publications, or get as close as we can. And then hopefully, in the exercises, we can ask you to expand on some of these results. Okay, I think this is a lovely framework to build training around, but it has the added benefit of once you start to try and recreate data from a paper, you start to learn to really critique the actual methods within some of these bioinformatics papers. Okay. So this is the course program which we will run. So I can say now we've met with the dean earlier this week, and we will be running some courses here at Rockefeller just after Christmas. So we're going to run this for the whole of the Rockefeller University just after Christmas, and we'll advertise this before. And it will be a first come, first served policy. But we'll also be running this as part of the graduate school. So if you're in the graduate school, you can attend these courses, and we're planning on doing this in March. And in there, you will be able to get course credits as well. Okay, so I'm just going to hop out of here again for a sec to take you actually. So this is the training page, and these are all our courses. Just wanted to show you some examples actually on them. So this is our reproducible R course. I try and describe within here how you can get going on the course yourself, rather than necessarily being in the class with me. And then in here it contains, hopefully reasonably consistently across them, all the code in the course, and then the HTML presentation or a single page version. Should all look fairly similar. So this is a course which isn't actually programming. This is our genomic file formats, just to tell you a little bit about file formats. And I just want to point out that they are carefully linked. So here we're telling you about genome reference in the file formats lectures, and later if you come to the ChIP-seq, somewhere here, we should tell you, yes, we provide a link which takes you back to that previous course. Which takes you back to that previous course. Similarly, just to show you, we also make sure that the data we're showing is available publicly, so you can go get this yourself, and I show you how to process it here in a different course. Okay, so these are all carefully linked. Great. So all this training, again, it takes some effort to try and maintain it all. And I would like to spend our time trying to develop new training and new methods to describe. So I try and put as much automation on this training as possible, so that, again, like with the pipeline, if I make a change to my course and it changes the results or an exercise answer, I need to know that before I stand in my course and try and present this to people. So I can use the same tools as I used for the pipeline. Every time I make a change, the course is all tested. All the exercises are evaluated using the test that package to make sure they give the same result. And as a benefit, at the end of all this, if it's successful, all my material is regenerated and put onto the website. Okay, so again, minimal intervention, hopefully allow us to spend more time doing more interesting things. So we want to teach this in the class and online, so I want to make all this material available online. And if you're using R Markdown, which you can tell I'm a big fan, really makes this quite easy. And from the same bit of code, I can make presentation, I can make the PDF handouts, I can even make interactive training. But what I would like to do with all this, when we get towards the end of the training, which will run next year, is to try and wrap this all up, surmise it into one big training manual. Okay, so I've just given you a demo. This is just an example here. I can show you in real life here. So this is just taking the same course material. I can wrap it up into one book, and then with this, I can have everything in one place, which I can search. I can decide I want to have a night mode. I can make it easy to read or really hard to read. I can do various things. I can also, I'm not going to click this because I need to install Counteract first, but this will take me to a GitHub repository, so you can actually, hopefully, and I really hope you do, contribute to this too, and you can actually, once we've compiled it, make a PDF. Right? So after this whole book is made, you may not want to look at it online, you may want to print it out and carry it around with you. You can also, I just spotted this, tweet, Facebook it, or Weibo it. But I really hope that at the end of all this, and working together with you guys and people who come to the courses, we can build a 2018 version of this. I think that would be a really fantastic end goal for all this training. So that's actually all I'm going to talk about today. I just want to say some thanks to Anthony, Jason, and Lewis, who's not here. But thank you for making me welcome in IT and helping me set up this core. My collaborators in Cambridge, Mark and Shamith, who have always helped me with training. And Zoe at GlaxoSmithKline and Ines at 7Bridge Genomics, who have worked with me on all this software and any publications we put together. Great, so thank you very much for your attention. Questions, I guess, right? Are there any questions?
So what does the SAI system mean? The SAI system. Your slides, there is a...
Which slide is it?
Oh,
He's asking about Travis CI, continuous integration.
Oh, sorry, CI, yes, continuous integration. So actually, I'm glad you asked that, actually. So, where did I have that? Yes. So, yes, continuous integration. So this is the idea of, you have GitHub where you put all your code, and then you may have something like, at the moment I use Travis to test on Linux and Mac, and I use AppVeyor to test this on Windows. So this is a fantastic tool. I recommend you look at this, because it's free if you're willing to make your repositories public. And it will install, so when I want to test this, it installs R onto a virtual machine, it will install all the libraries, it will then test my package, and then it will give me a report of what happens. It's fantastic. And this happens, I can set rules, but this happens every time I make a change at the moment. What's really good is if you take a branch from my pipeline, which I hope you do, and you make changes and add some great features, then this will test your branch and then give me the option, once it's all successful, to pull it back into the main repository. So I want to point out that although I'm using Travis at the moment, I really hope, let me point to him again, he's not here, so maybe this guy is doing some deployment for servers at the moment in IT, and I'm really hoping that he's going to introduce continuous integration into IT, so we have this locally, and that will mean I will be able to run much better tests. So, it's a free system, I can't run huge tests, but with one locally, I could really test it on lots of data.
So, I must admit, I've never Googled the term bioinformatics, but I was hoping it might include image analysis as well as just genome analysis.
Yeah.
Is that something you guys, because you haven't mentioned that at all, are you
No,
going
I
to
haven't.
help us with advanced image analysis?
I will endeavor to help you with advanced image analysis. As I have some experience in image analysis, if this looked like something which we would be a common data type in Rockefeller, which I think it might be, now people are doing this at such high throughput, we will also look to try and bring these skills into my team.
So if I can just add to that, there's a sort of an order of operating. The first thing is to really get genomics sort of an injection of sort of analysis support for that, and then probably next would be sort of more proteomic data, and I think image analysis would be in that sequence, but probably third in the sequence. But that to be said, now is a good time to sort of, if you have anything immediate to, before that all gets going, it's a good time to talk about it. So maybe you can talk for the next couple of months.
It's a great pipeline, and the vision looks really good. I know that you showed that a large portion of what you might project might involve custom programming, custom projects, and a lot of custom software design that can often sort of lead into experimental places that can take quite a long time with a lot of effort. Do you have any sort of ideas looking forward about how you might sort of stagger that, as well as sort of keep up the the basic pipeline package that you propose?
So you're talking about developing software for particular groups. I got to admit, it's not top of my set of priorities to develop software for particular groups. I'll always try and target something which is beneficial to more groups, more of Rockefeller, if I can. If I see a group wants to develop some software, which I think once we've developed it will be useful, then I'd be very keen to get involved. As our team grows, we may be able to take on more of these projects. And often, things like building databases, we may do this very particularly for one group, but what we learn from that may make it very easy or much easier to then apply this to other groups. So it's carefully thinking about the actual type of software or the type of project, I think, taking this one at a time. But just trying to do things which help as many people as possible.
I did have a question. First, thanks for doing this. You and your team, your vast team, I know it's housed in a large warehouse there in the IT pavilion. So genomics has certainly been the Wild West of analysis, and so certainly to bring it to a place where there's hope for reproducibility, so I just applaud it and thank everyone who's involved in this. And then on the training side, I don't think anything motivates someone to learn how to do something then will it get me something. So to use their data towards a publication, I think is a brilliant idea, and will get people to be trained. So along that line in your pipeline, I saw that you were able to export or download a lot of the graphics. Do you have sort of the publication package of the top 5 graphs that are going to be included in the publication that they're going to be able to use to submit into a journal? Yes.
That's a really good question. So I do. We have a course on visualization where we target, and I think Jason also has a course on this, ggplot. So it's a really popular tool. I will add a course on interactive visualization for the next round as well. But I think nailing things like ggplot is much better. I mean, all this interactive is fun, and it's useful to give this back to a user, but I don't think it will end up in a publication. And making something interactive, which also looks good when printed on paper, is quite tricky. But yes, we will try and target the most popular plotting packages. Any other questions?
Illumina likes to push base space for the downstream processing of sequences, and then there's also sources like Galaxy that have a lot of open source tools. How does your pipeline integrate or with those?
So there is some overlap with base space. So I actually wrote a base call QC in the last release of Bioconductor because I didn't feel like I was getting enough information back out of base space. For Galaxy, I think that's a different approach. Galaxy is more for users to be able to go, I think, and do a particular analysis. So if they really desperately want to align a data, you can go to Galaxy and run this. Us versus this pipeline versus, say, a Galaxy sitting in someone else's university, I think it's always going to be easier to be able to run this pipeline. It will be able to process the results quicker, and you'll certainly be able to get results to the system, or the pre-processed data to the system a lot easier this way than you would out of Galaxy. I mean, I wouldn't mind seeing if I can actually wrap this up into a Galaxy tool. I think that's perfectly plausible. But I see Galaxy more as a trying out bits of analysis than an actual solution. And I think the workflows in Galaxy, I would prefer something like this. I mean, we will actually, I haven't mentioned, one thing I haven't mentioned at all is Shiny, which is really another big thing about R. So we will start to look to try and make a Shiny interactive GUI version of this pipeline in the next releases. Thanks. Great. Well, thank you very much for coming.